So we're going to talk about recombinant DNA today. Really what we're talking about is a bit of technology that has to do with DNA. There are lots of examples of technologies that can be used to analyze and interpret and study DNA. We are going to look at two, but keep in mind there are tons more that we could talk about. The first one we're going to look at is genetic engineering. Maybe you've heard of genetic engineering before. The purpose of genetic engineering is to make recombinant DNA. So recombinant DNA is the result, it's what you get when you manipulate or you engineer genetic material. In a lot of cases, what's happening in reality in genetic engineering is that we insert a new gene in an organism. There are other applications of genetic engineering, but this is a pretty common one, and it's what I'm going to describe here. So we're going to manipulate genetic material by taking a gene from one organism and inserting it into another. Now we can do this because, like we saw a few lessons ago, the DNA code is universal, so all organisms are using the same code, which means I could take another organism from the same species, or I could take a sequence of DNA from a different species, and it should make the same protein in the long run, no matter which species I put it in, because the code is universal. So if we want to do some genetic engineering, there are a few things that we need. First, we need restriction enzymes. Restriction enzymes have as their function to catalyze the cleavage, or we could say cutting, of DNA at precise nucleotide sequences. So not just anywhere, they don't just randomly cut the DNA, but they look for a specific sequence to bind to so they know where to cut. So if I look at the little picture on the side, each one of these little red, weird-looking, toothy Pac-Man guys is a restriction enzyme. This is not what they actually look like. This is just a picture so that maybe you'll remember that they're for cutting. They are, aren't they? They're adorable. And if you look where the arrows are pointing, the restriction enzymes will cut kind of like this. Now, they aren't just going to cut anywhere. They are looking for a very specific sequence of nucleotides. If you look at the white side of the picture, the letters, I'll show you on the two sides. It goes T, T, A down here, and A, T, T up here. So that restriction enzyme was looking for AATT. On the black picture, you can't see the letters because, of course, it's colored black, but the shapes still match up. And we'll see in the picture down below, but if I take a peek up here, I have the same nucleotide sequence, A, T, 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 A. Now, why are there two pieces of DNA? Well, that's the other part of this point. There needs to be a donor and there needs to be a host. Something needs to donate this segment of DNA that I'd like to insert, and something needs to be the host. I need an organism to put it into. So we're just going to say in this picture that this one is the host and this one is the donor. So I want to take a piece of the donor's DNA and add it to the host's DNA. So I start off by cutting both organisms' DNA with the same enzyme at the same sequence. Now it's important that I cut both of their DNA. If I don't cut the host's DNA, there's nowhere for a new piece to be inserted. If I don't cut the donor's DNA, I don't have a piece of DNA that I can donate. Now does anyone want to ask anything about that point? What I end up with, if I use those restriction enzymes, are fragments. So if you take a look over here, I have some fragments. The restriction enzymes have cut the double-stranded DNA helix, leaving me with a few DNA nucleotides that don't have a partner anymore. Sometimes we'll call these little fragments cohesive or sticky ends. They're not actually sticky, like if you touch them, they're not going to stick to you, but we call them sticky ends because we have some unpaired nucleotides, and DNA does not like to be unpaired. DNA exists as a double helix. So if I leave two complementary sets of unpaired nucleotides lying around, there is a high probability that they will join to each other. DNA wants to be double-stranded. So if I look, I have TTAA down here, and very conveniently, I have the exact complement to that on that black piece. So I cut the two pieces of DNA in the first step so that they had complementary sticky ends. That way, if I put them together in the same container, they should attach to each other. Now, the last step to this, to make sure everything stays stuck together, is that we need ligase, the same type of enzyme that we talked about with DNA replication. Ligase is like the glue. Maybe you remember us watching that little video. It would come along and help join together the sugar phosphate backbones on either side and make sure that all of those new nucleotides are attached to each other. Now, this is the very basic way that genetic engineering works. Recombinant DNA is what we're going for. It's the goal. It's what I want to get. Restriction enzymes are what we use to cut things, to give us fragments. Ligase, which is the last one, we generally only use in the host. I've taken a piece of donor DNA and inserted it into the host. I'm not inserting something back into the donor's DNA, so we don't typically use ligase on the donor. Now, if you were to be asked about genetic engineering, you would definitely need to know these two enzymes, and you would definitely have to understand that those fragments, those sticky or cohesive ends, are complementary to each other. Now, does anyone want to ask anything there? There are some really cool applications of genetic engineering. Maybe you have heard of some of these before, but there's this cool gene called the antifreeze gene. Some fish have it. It lets them survive in really cold water. And people have been experimenting trying to put it in wheat and tomatoes and other types of plants so that they can be cold resistant. So that's an example of something that genetic engineering could be used for. Now, another technology, and we will look at this one pretty specifically, is something called gel electrophoresis. If you ever wanted to talk about gel electrophoresis and you could not remember the word electrophoresis, you could just say DNA analysis on gel and tell me about it and not use the word electrophoresis. In gel electrophoresis, we're going to take samples of DNA and analyze them to see if they're similar or to see if they're different. I'm going to give you a worksheet in a little bit where we'll continue this analysis, and you'll see that some of the reasons we would want to do this would be criminal investigations. I have a sample, I want to know which suspect left the sample. For identifying people or doing paternity tests, so figuring out who the biological parents of a child are, we would use something like this potentially. Now, in gel electrophoresis, we're going to separate molecules according to their mass and their charge. So here's how it works. We are going to make some DNA fragments using restriction enzymes. Same kind of enzymes we would use for genetic engineering, we can use for this. Restriction enzymes, like we described on the last slide, cut DNA at a specific sequence. So I could take a sample of someone's DNA and add some restriction enzymes to cut it up into pieces. If I have the same DNA as someone else, then the restriction enzyme should cut our DNA up into the same number of pieces, and the pieces should be the same size. So let's go. I'm going to use my triplets here as an example. Theirs should be the same, or really close to the same. But if I were to compare me with one of them, there should be differences, because we would assume our DNA is not the same. Now, once we have fragments of DNA, we're going to put them into the end of a gel. So if you look up there, there's this little sequence of 6 pictures. There's also a pretty good picture of this in your textbook in the section on DNA analysis, if you don't want to draw it here. The rectangle is like a little box that's full of gel, a little bit mushier than actual Jell-O, but more or less that consistency, this gel. And there are holes or wells at one side of the box. In each of those holes or wells, we can insert this sample of DNA that we've cut into fragments. So the thing that looks like a little syringe is actually a micropipette, and we're just squirting our samples into those wells. Now if you look, okay, the first one, then you can see it's full, and we put it in the second and the third one. Then we apply a current. So starting on picture 4, there's a plus on one side and a minus on the other side to show us that a current is being passed through this gel. Now, because of the polarity of these DNA nucleotides, they will be attracted by this current. And once the current is passed through the gel, the smallest fragments move faster. So if I look at the 4th or the 5th and then the 6th picture, I would interpret that the fragments that stayed near the beginning of the gel were bigger fragments, and the fragments that moved farther in the gel were smaller fragments. The result of doing gel electrophoresis is forming a genetic fingerprint. Kind of like how you have fingerprints from your actual fingers, you can make a genetic fingerprint by using DNA analysis like gel electrophoresis. The picture on the right is a picture of an actual gel that has been run. It has a whole bunch of wells along the top, so instead of just 3 samples, there are about a dozen up there. If you look, they all have little bits of differences, but I'm just going to maybe highlight these 2. Those 2 columns, those 2 samples, once the current was passed through the gel, made the same bands at the same places on the gel. If I can look at 2 samples and see that they left behind the same fragments, I could conclude that they had the same DNA. So if I were to look at those 2 samples, I would say either they came from the same person, or maybe they came from identical twins or another set of identical multiples. But that's how we can identify a person, by matching their DNA sample run through a gel with another DNA sample. Now, does anyone want to ask anything there?